Hello everyone, and welcome to another webinar organized by the ANT Neuroeducation. I am Giulio Di Cosmo, an application specialist in ANT Neuro, and I'm very happy today for hosting today's webinar on atypical neural processing in 22q11.2 deletion syndrome and schizophrenia towards the neuromarkers of disease progression and risks. With me today, the presenter is Professor Sophie Molholm. Thank you to be here. She is a cognitive neuroscientist at the Albert Einstein College of Medicine, with appointments in the Department of Pediatrics, Neuroscience and Psychiatry, and Behavioral Sciences. She uses multimodal brain imaging, psychophysics, and standardized cognitive and clinical assessments to probe the brain processes underlying perception and cognition in neurodevelopmental and neuropsychiatric conditions as autism and schizophrenia, as well as the genetic disorder, like the one just mentioned, and the Rett syndrome. Just a little note, after the presentation, there will be a demo session recorded by our applications team, and then we'll follow the Q&A session. That being said, also, for all the attendees, you can see a chat box on the right. During the presentation, everyone is muted, so there is this chat box. Please feel free to write all the questions that you have in your mind, and then we will discuss them during the Q&A. That being said, Professor Molholm, thank you again for accepting the invitation, and we are very much looking forward to learning more about your research.
Thank you for the invitation, and it's my pleasure to be here. So today I'm here to talk a little bit about 22q deletion syndrome and how we use electrophysiology to try and really probe and discover neuromarkers of disease progression and risk. And so I'll begin by talking about the 22q deletion and the phenotype that's associated with it. And then I'll go through some findings that we have, really focused on sensory processing, auditory sensory processing, as well as higher order executive functions, with an emphasis on response inhibition. So 22q is a representation of the deleted chromosome from chromosome 22, and this, as you can see, so the deletion can go from A to D, which is a 3 megabase deletion, or it can go from A to B. Those are the most common deletions, and the larger deletion is associated with greater impact. So, but I won't go into too much detail about that. Also, perhaps relevant for you to know is that there are mouse models of 22q deletion syndrome, and most of the mouse models mostly mimic the 1.5 deletion, so the smaller one, but they're currently, or more recently, a model that mimics the larger deletion has been created. So over the past several years, what is the prevalence of 22q? Well, it's not a common condition like autism or idiopathic schizophrenia, but recently it's reported as being as frequent as 1 in 1,500 participants in the general population. So not super unusual. This was from a study, the SMART study, which found that the microdeletion is much more prevalent than had been previously thought. 22q is characterized by cardiovascular abnormalities, immunodeficiency. Sorry, my slides aren't advancing here. So let me see. hypocalcemia and other endocrine issues, cleft palate and hypernasal speech, small stature, and characteristic facial appearances. Now, so here are some examples of individuals with 22q, and you can see some of those characteristics in the face and the cleft palate characteristics. But what I'm really going to focus on and why we're interested in 22q is because it's associated with cognitive impairment and more relevant psychopathology. And the psychopathology is very similar to what we see in schizophrenia. So the incidence of psychopathology in 22q is as high as 50%, so seen in 50% of people with this syndrome. And schizophrenia, which is seen at about 1 in 100 in the general population, is seen in about 30% of individuals with 22q. So this makes it a really good genetic model to try and interrogate the underlying neurobiology of schizophrenia. It also means that families who have a child diagnosed with 22q, this is one of their chief concerns. Is my child going to go on and develop a psychiatric disorder that's as severe as schizophrenia? So with regard to cognitive impairments, the majority of individuals with 22q have intellectual disability. Here we can see a comparison between the distribution of IQ in individuals with 22q versus the general population. And what you can see is that the distribution of IQ is shifted to the left compared to the general population by almost 2 standard deviations. So with a mean of 72 compared to the standardized mean of 100 in the generalized population. The IQ isn't the only type of difference that these individuals struggle with cognitively. There are mild to moderate delays in all areas of development, including gross and fine motor development, general cognition, speech and language, already present by 2 years of age. And you really have this buildup of evidence of different cognition as the child gets older. So 3 to 6 year olds will show mild to moderate delays in all areas of development. And that will become more evident as they start going to school. And then when you get to early adolescence and on, you begin to see really divergent cognitive trajectories. So by that time, IQ is relatively stable, but there may also be appearance of cognitive decline and verbal decline and the beginning of evidence of psychotic symptoms. The psychopathology follows a very interesting trajectory. So here's the rate of psychiatric disorders in 22q. I hope you can see my mouse here. And so the earliest emerging differences are in anxiety disorders, and those are prevalent throughout the lifetime. And then you see different trajectories for things like psychotic symptoms, which tend to onset in adolescence and really increase into adulthood. So as I mentioned before, we see this as a unique model to investigate the neurogenetics of both intellectual disability and psychosis. Today, I'll really be focusing on psychosis and making comparisons with what we see in individuals with schizophrenia. So I'm going to be talking about electrophysiological data. And so here's our general setup. So we put a cap on individuals. We do high density recordings with 64 channels in a sound attenuated booth. Here is this is not a child with 22q, but this is a child actually with another syndrome. And this is Ana Francisco right here, and she's responsible for these the vast majority of the data that I will be presenting. And this is Elise Taverna. They're both PhDs and with expertise in electrophysiology. Sorry, I'm getting stuck with advancing the slides, but I got it. So this is just a recording of the EEG. We're recording from 64 channels. However, I will be showing you single channel data and usually just one waveform. So let me just break it down for you very simply here. On this axis, we have amplitude of the signal, and then we have time here. We're usually talking about milliseconds, and then I will talk about components, and these components are labeled. The labeling is a little bit arbitrary, so I won't go too much into how they're labeled, but you can see there's a sequence to it: P1, P2, P3, N1, N2. In this case, the P's indicating positive response. Right. So that's just Dr. Francisco with a participant in the lab. And now I'm going to show you some data from an auditory paradigm that we ran. And in this paradigm, the data that I will show you include data from 26 controls and 26 individuals with 22q deletion syndrome, all matched on age, more or less matched on sex. And however, we have differences in both IQ and working memory between these groups. And this is just going to happen if we're looking at this population and comparing them to a healthy control group. And then we further, because we're so interested in psychosis and 22q as it relates to schizophrenia, we divided our sample of 22q participants into those with psychotic symptoms and those without. They weren't currently necessarily experiencing those symptoms, but they had experienced them and they reported them. And 15 of our participants were with psychotic symptoms and 11 without, allowing us to really have a nice look at how the data might differ dependent on whether they're showing psychotic symptoms or not. And here you can see that these groups were very well matched. The IQ of the ones without psychotic symptoms was about 74, and it's about 70 for those with symptoms. So a small difference there. And working memory is also a little bit poorer in those with symptoms. But even the group without psychotic symptoms is lower than the general population. The paradigm that we used is an oddball paradigm where we just present auditory stimuli in a series of tones, and we measure the brain response to those tones while participants are engaging in another task. Here, often just watching a silent movie. And the beauty of this paradigm is it allows us to look at the brain engaging in sensory registration and also higher order cognitive processes of sensory memory, even though participants aren't attending, so it's appropriate for any age and any functioning level. And what we're going to focus on here, what I'm going to focus on, is the auditory N1 response and the mismatch negativity. The N1 response reflects activity in primary and secondary auditory cortices. So it's really a nice way to see, is sensory registration occurring in a typical way? The mismatch negativity is a response that's involved in comparing current auditory input with a transient memory trace of ongoing regularities in the auditory environment, allowing us to look at sensory memory when there's a perceptible change between the stimuli. So in this case, the deviant stimuli were longer. There's a mismatched negativity response. And then for this paradigm, we presented stimuli at different stimulation rates under the idea that we could increase demands on the working memory system by lengthening the time between stimulus presentations. And first, I'm going to talk about the N1. You can see that this is the auditory N1 response here. And if we just compare the N1 of our healthy control sample and the 22q sample, you see that the response is a little bit larger for the 22q group. So there's an enhancement of the response. This is actually in the opposite direction to what we see in schizophrenia, where the basic sensory processing as measured by the N1 is smaller. So what happens when we divide our 22q sample into those with and without psychotic symptoms? And here what you can see is this really interesting pattern where the 22q without psychotic symptoms show a much larger response now, whereas those with psychotic symptoms show a response that really looks comparable to the control group. And this is a little bit surprising. But it's also what's seen both in animal models of 22q and in prior studies, which is this increased sensory response. So it seems like an increased sensory response may be a signature of 22q deletion syndrome and reflect very basic auditory processing differences. And then what we hypothesize is that for the group with psychotic symptoms, there's a diminishment of this response, just like you see in schizophrenia. But because the baseline is much bigger for the group, that ends up looking like the control participants. And so really we're thinking, we think of this as this kind of two opposing mechanisms. One related to the deletion, resulting in increased early sensory responses, and another associated with the presence of psychotic symptoms, which has as its outcome decreased sensory responses. Now moving on to the mismatch negativity response, and I apologize, I didn't, oh no, this is fine. So here you see the dark trace is the response to the standards in the oddball paradigm, and the stippled trace is, the stippled trace is the response to the deviant stimuli, that is the stimuli that were longer in duration. So the brain registers when the response is different, and we subtract the deviant from the standard, and that gives us what we call the mismatch negativity response. So here's that difference wave for the control participants and the 22q participants. And you can see that there's virtually no difference between these groups. That's quite remarkable, actually. But then when we go and let's see what happens when we break it up in terms of psychotic symptoms versus no psychotic symptoms. And here you see something quite different. So the blue trace is our controls, and the maroon purple trace is our 22q participants with psychotic symptoms. And they show a diminished mismatch negativity that's consistent with the schizophrenia literature, where mismatch negativities are smaller in schizophrenia compared to controls. In contrast, if we look at the group without psychotic symptoms, if anything, the mismatch negativity is larger. But the main thing is to note that the response is smaller for the group that's more similar to schizophrenia. So just summarizing with pictorially, we see that the 22q deletion syndrome have a larger N1 compared to controls. And if you break the 22q group into positive and negative, this greater response is really only seen for the without psychotic symptoms group, and then we see a diminished response for the other group. And for the mismatch negativity, there's no difference in the magnitude of the response. Just looking at the group level, when we break it up, we see that there's a diminution of the mismatch in the group with psychotic symptoms. So now moving on to, so there we're talking about sensory processing, but what about, and we showed different findings, and we show that this really differs depending on whether you have the presence or absence of psychotic symptoms in this group. When you have the psychotic symptoms, it's looking much more similar to people with schizophrenia. What about when we go on to higher processes, so what are called executive function processes? This is what you would really expect to be disrupted in a group with lower IQ and impaired working memory and psychotic symptoms. So the way we look at this is with a really simple paradigm. We're always going for relatively simple paradigms here to make the tasks relatively easy. We are working with a compromised population, so that's important. And so this is called a go, no-go task. And what we do is we simply present images, and one after another, relatively rapidly, and participants press a button to each stimulus as it comes. So here's a screen, an image, they're supposed to make a button press. Here's another image, they're supposed to make a button press. But if an image repeats immediately, they're supposed to withhold the response. That's called a correct rejection. It's actually very difficult to do because you get in this rhythm of pressing a response, and then suddenly you have to withhold that response. So what we look at, we could look at basic visual processing. I'm not going to talk about that today. But we look also at the P3 response, which you see to those is much larger for the withhold responses. And to error-related activity called the PE, which occurs when somebody presses the button, even when they're supposed to withhold that response. So it reflects the perception of an error, registering that you've made an error and you're aware of it. That's important because you can only correct your behavior if you're actually aware that you've made a mistake. All right, so here we looked not only at 22q, but also at individuals with schizophrenia, because we wanted to now be able to directly compare, not just with the literature, but in our own data, how do these modulations for the 22q positive versus negative group compare to individuals with schizophrenia. And we needed separate control groups for each of those because the age was a little bit different, but that's not so important here. And then for the 22q group, we again broke them into those with positive symptoms and those without. And by the way, when I say positive symptoms, and I should have explained this before, we're talking about positive symptoms that are associated with schizophrenia, such as paranoia and delusions and hallucinations. Schizophrenia is also associated with negative symptoms that I'm not going to talk about here, that we didn't use to classify our groups. First, I can just show you the differences in IQ. So if we look at our control group compared to the 22q group, you see there's a significant difference, and that's present whether or not they have these positive symptoms. Then taking a look at our schizophrenia group, we also see a higher IQ for the healthy controls compared to the patient group. That's expected. Then in terms of performance, because here people are actually performing a task, what we see is that performance is not hugely different between our patients and controls, but it is significantly different, both for the 22q group compared to controls and for the schizophrenia group compared to controls. And D prime is a measure of performance accuracy. So did you withhold the response when you were supposed to hold the response, withhold it? We also looked at reaction times. And here again, we see small but significant group differences. And then if we break down our performance measures, just focusing in on our control group and schizophrenia and the 22q groups, you can see how that breaks down. So there are some significant differences there. That with schizophrenia differing with longer reaction times compared to the other groups, but they're also older, so that's not surprising. And then the 22q positive symptom group differing significantly from the control group in reaction time, but not the 22q without symptoms. And I'm going to be focusing on two responses here, as I mentioned before, the P3 and the PE. This is what they look like generally. And the P3 is the response that you get when you make a response, when you withhold your response to the repeated image. And the PE is the response you get, the error response, when you make an incorrect response to a repeated image. And so looking at the P3, we can look at topography of the response. So these are topographic maps showing the relative distribution of activity over the scalp. Here's our control group, and here's the 22q group. And you can see there's small differences, but not huge. So nothing too prominent there. And so what we're doing is I'm going to show you waveforms that contrast our difference wave between the response to the hits and the response to the correct rejections. There's your P3. We do a difference wave and you get a single response. And here's the P3 for the control group. It's largest. For the 22q group, it's clearly smaller. And for the schizophrenia group, it's the smallest. What about when we break our 22q group into the two different groups? Then what we see is that the 22q participants without positive symptoms for schizophrenia look quite similar to the control group, but those with positive symptoms, so symptoms of schizophrenia, are looking very similar to the schizophrenia group in terms of their P3 response. Then focusing in on the error response, you can see that the most robust response of registering that you've made an error is seen in the controls for the 22q group, and then next in the control group for the schizophrenia patients. But when you look at the two patient groups, you're getting very small responses in terms of registering the error response. And if we break it down and now just compare our control group to our three patient groups, the two 22q ones plus the schizophrenia, you can see that across all of our clinical groups, there's an impaired awareness or registration of making a mistake. And what that means is that that information is going to be less available to kind of update the model and say, pay more attention or something like that. And so just to summarize, the P3 was only reduced in the presence of psychotic symptoms and therefore may be a marker of disease severity. And the PE was equally reduced in schizophrenia and 22q, regardless of the presence of symptoms. And so from this, we conclude that 22q seems to be associated with increased sensory responses, particularly in the auditory domain. We've also looked in the visual domain. There are differences there, but they're not as prominent. And that in the sensory domain, responses of those with 22q and psychotic symptoms approximate the responses of those with schizophrenia. And this suggests that the mere presence of a couple of psychotic symptoms is enough to change brain processing and make it look more like somebody who has chronic schizophrenia. The response inhibition findings are particularly relevant in that they point toward a potential marker of risk for schizophrenia. That is, it's present whether or not the patients have shown symptoms. So just the mere genetic risk of getting schizophrenia seems to be reflected in the error response, meaning this could have clinical implications for early diagnosis or possibly as an outcome measure for different treatments. So with that, I would like to end by just thanking all the participants that contribute to these studies. It's difficult for a lot of these participants to participate. There's some resistance to coming in and having a cap put on your head if you have psychotic symptoms and paranoia. But Ana Francisco, who you can see here at our 22q clinic, really made a point of building relationships with our participants. And that has made all the difference in their willingness to come in and sit in a cap and engage in these studies so that we can try to learn a little bit about brain function and dysfunction in these groups. And then, so there's Anna with a different hairstyle, and my colleague John Fox, who's also been involved in this research, and then also Dawe Horswit, who was the main technician collecting these data and doing a lot of the pre-processing. And with that, I will end my presentation.